Finding a new species in paleontology is not far off an everyday occurrence. It's pretty easy. Finding a specimen that seems to possibly solve a century-old mystery, though, that's something less common. Hello and welcome back. Or welcome for the first time, because I am astonished by how quickly I'm getting more people coming and sticking around. Although that said, I have noticed that over 90% of the view time is by people who aren't subscribed. Hint, hint. Anyway, it really is lovely to have you all, and thank you so much for all the encouraging comments and even all the comments encouraging Lucy to come back, which is going to happen today. Today, though, I promised you some really strange fossils. And this is, well, certainly true. And potentially it even solves a mystery that has been baffling many of my colleagues for quite a long time. So in paleontology, we generally find that most of the fossils we discover you can immediately put into a major group of animals or plants or whatever it is. That you know you've got brachiopod or trilobite. Even if it's a weird new species, you know where you can put it, and you know why it's weird. But there are a few groups of fossils, and there are still a few left, which have been enduring mysteries. There have been classic examples of these, like the conodonts, tiny little tooth-like elements, which were used for dating rocks, but nobody knew what they were, until a complete specimen turned up in a drawer in the Geological Survey in Edinburgh, which had been there for something like 100 years, and nobody had noticed that it was the entire animal with the teeth in place, until Ewan Clarkson did. So these things get solved eventually. Or sometimes it's a new specimen, like Macaridians were enigmatic plates that you find occasionally articulated, mostly as isolated little fossils in mudstones around the world. And then in Morocco one turned up partly disarticulating, but with the soft tissues preserved, including the gut and the little bristles, and this revealed that these things were worms, polychaete worms. So occasionally you need a key specimen to help you work out what problematic things are. And there are still quite a lot of mysteries. So we have things like chitinozoans, which I'm sure I'll talk about at some point, because they're weird little microfossils, but there's a big debate over them at the moment. And then there are less well-known ones, but still equally significant in some ways. So today I'm going to be talking about banded tubes. If you spend long enough looking for graptolites in the shales of the Welsh Basin, so out in the middle of Wales or even in the borders, you occasionally find layers which are covered with bits of debris. Indeterminate bits mostly, little flakes of carbon and little filaments and fibres. And these fibres turn up abundantly from the early Silurian, but mainly into the middle and then the late Silurian, and also into the early Devonian apparently. Well, I've not found them there, but they do have this fairly wide range, Silurian into the Devonian, which coincidentally is also coinciding with the earliest fossil land plants, aside from isolated spores. They turn up through the Silurian into the early Devonian. I think there are some records of similar things from the latest Ordovician, but what you're looking at is tiny little filaments. And these are often found in dense clusters, so little bundles of them, all a bit ragged at the edges. And they are completely mysterious as to what they are. Many things have been argued, and in particular the graptolite specialists, who tend to find these far offshore in what we call hemipelagites, so sediment which has filtered down through the water. Apparently. I think it's actually a lot of this stuff is being washed in and sort of along the sea floor, but still it's a long way from the shore and in very fine sediment, and you get layers covered with these little filaments and fibres. And graptolite workers have tended to call these chitinous hydroids. So they look a little bit like the little rods and branches that make up hydroid skeletons. And it's possible, but you don't tend to see any real hydroid features. You don't find the little cups where the animals were living and so on. The other main school of thought is that these are something to do with land plants, and that in fact what we're looking at are the water-carrying vessels, the tracheids, of land plants that are being washed out into the shallow waters and disintegrated, and all we're left with are these little bundles. And so because of this, and it's potentially important for understanding early land plant evolution, the palynologists, the paleobotanists, have put a lot of effort into these, including electron microscopy work, and they have found that they have a very distinctive structure. So there are annulations, particularly on the inside of the tubes. These things have sort of thickenings of the wall going on the interior, and they appear to differ significantly from the known tracheids of the early land plants. And the people who did this study concluded that they were probably some sort of heterotrophic fungal-like organism instead. They were for a long time also referred to as a nematothalus, which is unfortunate because that was by association with other little cuticular fragments. It's nothing like tubes, little flakes of organic material with a reticulate patterning on them. And these were long thought to be also related to either algae or land plants, and maybe they were different parts of the same organism. Although the consensus seems to be now that they are actually quite distinct. You get them very commonly co-occurring, but not always. And there's a recent paper, again by Nick Butterfield, who turns up a lot in this channel, showing that they are almost certainly some sort of red alga. So that's the cuticular bits. And the filaments, don't know, nothing to do with us. Understandable. So what we really need is some consistency in what our understanding of these filaments are. Being able to rule out multiple other different types of filaments, because if you just look for little tubular organic microfossils, there are loads and loads of them, and you have to be sure you're looking at the same thing. But they're all sort of problematic things. There's nothing which we can point to and say, here is a fossil which is made of little organic filaments, and it is almost certainly disarticulating to make that. Of course, that's what we really want, isn't it? Well, just so happens that might be exactly what we have. This is something we've actually had for a few years from a site quite close to our home. It's walking distance, and it's an interesting place. It's in the middle Silurian. It's out in the middle of the basin, but there's suggestions that there was relatively high sea floor there, so it's a topographic high. It was more aerobic than some of the surrounding areas. We have sponges there, we have crinoids, complete crinoids. It's quite a diverse little fauna. And we also have some things that are problematic. And these things are difficult enough that I think this is going to be a two paleontologist problem. As many of you are aware by now, I had the eminent good sense to marry another paleontologist who wouldn't complain when I filled the house full of rocks. Meet Lucy.
Hello everyone, I'm Lucy. I am a paleontologist. I have a house full of rocks. I worked on quite a lot of fossils over the years. I've done quite a lot on graptolites, my favourite fossil. Joe gets me into doing sponges occasionally. I've done fossil worms. I work on the Castle Bank and other sites with exceptional preservation, especially in the Ordovician. But what I started out doing in my PhD was Silurian graptolites, mostly in the Welsh Basin. So I didn't collect just graptolites during my PhD, that was my focus, but I would keep whatever I found. And I would find these sort of odd little black thread things that I didn't know what they were. And my supervisor would say, oh, well, they're chitinous hydroids. And oh, all right, chitinous hydroids, that's fine. And they were very small bits and I couldn't really do anything with them, so they were just a little curiosity, really. But it seems as though we've got much better material than what I found during my PhD.
We certainly have. So we've got actually several specimens of these things now, but this is the first one we want to show you. There's a nice big graptolite on the slab, obviously, but next to it there's a little centimetre long dark streak you can just see there. And that is what we're going to look at first. First thing to notice, on this surface there are lots and lots of different things. So there's a graptolite running north-south up the middle here, and that is crossing some of these tangled bits of filaments, which is a good start. But then if we move across to the side, we have here the base of the thing that we're looking at. And right next to it there's something else which we'll talk about in a minute. But let's have a look at this thing first. I want to zoom or move up it. You can see it's elongate, it's made of lots and lots of little black fibres. We'll zoom in a bit. And it comes to a sharp end with little fibres sticking out of that end.
So let's have a look at the internal structure of this thing a bit. We've got these two dark lines here, so what do they look like in close-up? Let's focus. And they do seem to be bundles of parallel fibres, and then there's lots more fibres running parallel to them. The whole thing seems to be these parallel fibres. Are these banded tubes, Joe?
For that, we would really need to see them in SEM to make sure that they do have this internal structure, if that's visible. But they certainly look like it. We have the same arrangement of these little dense bundles and then isolated tubes. And if this was washed around on the seafloor, it would disintegrate to form something that looked exactly like the debris that we see all over these rocks. So I'm sure that is the same thing that we've been calling banded tubes. Whether it's exactly the same thing that the paleontologists find is another question. There may be different types.
So we'd really need to put this in an even better microscope and see if we can get even more detail to verify that.
Yes, but we can say that this is an example of the thing that many people have been calling banded tubes or chitinous hydroids. But is this a whole fossil? It's actually, there's a couple of points of interest here. One is you'll notice that over the fossil itself there's this sort of brown stain which is different from the surrounding matrix.
Okay, so do you think that would be soft tissue?
It looks like it, doesn't it? It looks like organic material. It's separate from the tubes themselves. It's quite diffuse, but it's there. It's a definite colour. There is a clear difference in this sort of brown colouring over the surface of the thing, then a sharp edge, and then it goes into normal sediment. And this is not what you normally see with these tubes.
No, you've also got these little black dots at places over the surface. Is that significant, or could it just be random little black dots?
Yeah, there are little random black dots over the surface of the rock nearby as well.
So probably just little flakes of carbon. Yeah, there's some just to the left here.
Yeah, and we know that this thing was full of graptolites, which were, shedding bits of carbon all over the place by the looks of it.
Messy creatures, what can you do?
You can't take them anywhere.
Yeah. Okay, well, next question. Could this be something like a fecal pellet, that something has eaten whatever was making these tubes, and this has been excreted out the other end?
Yeah, that is always something to consider. In this case, I would say not, because it is the same density across the width of it.
That's true. And none of the fibres seem to be obviously broken, which would be unusual if the thing had been eaten.
And they're all very neatly aligned.
Mm.
So if this was a pellet, you'd expect firstly the middle to be really thick compared with the edges, and it should be made of mangled mass of these things. Whereas what this looks like is something flattened. It's on the surface of the rock, and it's got this sort of bundle of spikes sticking out the end. So some of those fibres extend and then sort of flare out from the tip, but not from the sides. It's just from that central part.
Yes, that's an unusual sort of structure. It does look rather like something like an algae or land plant. I suppose the next question is this the whole thing, or could it be a torn up part of the thing?
Yeah, okay. We do have clear endings, so this top end seems to be sort of rounded, and then you've got this structure of axial stuff going out the top. The other end is a little bit less well preserved, but you do seem to have something like a sort of fairly sharp tapered point.
It does seem to be pointy, doesn't it?
Yeah.
Yes, that's not as clear as you might like. I suppose the other thing is, do we have any other specimens of this? Is this something consistent?
Absolutely. When you see something like that, you're not quite certain what it means, you look for more. And luckily, we do have more. Okay, so on this slab, there's actually a couple of things we want to show you. One of them is this one here. It's quite small, another centimetre-sized one, again, very similar size to the previous one. We'll also have a look at that one at some point as well, but this one first. So if we're looking for consistency, this isn't bad at all. We've got this narrow rounded base, we've got obvious longitudinal fibres going up. In places there's denser patches where they're more bundled, and then you get to the top, and if we zoom in there, then it's slightly difficult to see because there's this sort of brown stain over the surface around it, but there's also the pale prep marks where I've sort of cut out the end of it. But here you can see that at the top of the sponge there are indeed these black fibres extending past the margin again.
Oh, you just called it a sponge. Did you mean to do that?
No, I didn't, did I? Whoops, sorry. The top of the fossil.
Let's not jump to conclusions, because I still think that alga is actually quite a good option at this point.
Yeah, absolutely. Okay, so why would alga be optional?
Well, you get taxa like Medusigraptus, which at least partly consist of bundles of parallel filaments. I mean, not the whole thing like this. And this does seem to be the whole thing. We've got two that are reasonably consistent. So I think that a structure full of bundled parallel tubes is actually fine for alga, or possibly even for land plant if those are the tracheids, the water conducting vessels.
But is the shape of this thing convincing for an alga or land plant? It's doing nothing to spread its surface area out.
It is quite.
It's a
fusiform, isn't it? It's thicker in the middle and thinner at both ends, which is slightly odd for an alga. I wouldn't say it was impossible.
Well,
especially if there were perhaps big light gathering leaves or something that haven't been preserved.
But we are seeing this sort of relic of soft tissue remains, this sort of brown stain over the surface of the fossils, which suggests that we are seeing some quite soft structures as well as the tubes.
Oh yes, but if it is alga or a land plant, it would have been transported to where we found it from somewhere else, and anything really soft and delicate, like a leaf equivalent, might have just been destroyed during transport.
Possibly, yeah. Okay, so another thing, if these were some sort of tracheid-like structures, if we're going for the land plant option first, wouldn't you expect those to be in a bundle in the centre?
Not necessarily, because you think some plants you got the sort of soft pith in the middle and the woody bit is around the edges. And this isn't quite woody enough to be woody if it's a plant, but there's not necessarily any problem with it just having parallel bundles of fibres interspersed around it. It wouldn't have been a very big plant, so it's not like it was trying to support itself on a 100-metre tall stalk or something.
Okay, so why would you get bits sticking out the end? Some of these fibres actually extending beyond the edge of the plant.
Well, if that had been where the fibres were going into the leaf equivalent, you might expect that. You might expect that some of them would just have gone into the leaf a little way and got torn off.
Interesting, interesting. So,
or alternatively, we're looking at it the wrong way up, and those are actually little rooty equivalents.
Could do. So what we would ideally need is another specimen that is obviously the same sort of thing, but a bit different.
That would be really helpful, wouldn't it?
Would it?
Yes.
Oh.
Have we got one?
You never guess what I prepared earlier.
Oh!
Okay, this one is a very different shape to start with. It's much broader, it's got a very wide rounded base, and it's much wider at the top as well. So it doesn't look immediately similar. So why am I saying it's the same thing? Or similar?
Why are you saying it's the same thing?
Yeah, good question. Let's have a close look. So if we zoom in, well, if we move upwards first, to start with, we're just not seeing these longitudinal little filaments at all. But then when you get to the top, stop there, and yeah, so let's focus in a bit more here. Then you can see coming out the top of it, in the middle, is a bundle of filaments, or an array of these little black filaments, which look identical to these things that are being washed around, deposited in these layers. And if we can zoom in on this part, below the part where they're coming out, then you can see that these filaments are continuous with the ones sticking out. And they carry on as a little, not quite a rope, it's not as dense as that, not quite even a bundle, but there is an array of these filaments going down into the centre of this object.
Okay, I can see those, and those do look similar to the longitudinal filaments of the other ones, but the overall structure of this thing is a bit different, isn't it? If we go to here, for example, you've got some vertical filaments, yes, but you also seem to have filaments going horizontally. You've actually got more of a grid structure here. I think this might be something a bit different.
I agree, it is a bit different. But it's another specimen where we're seeing these black filaments in a very similar sort of array. So you've got the longitudinal bits going up the centre, you've got smaller and finer ones around the edges, and yes, there is a grid there, bits going in different orientations as well.
Okay, so looking at this, I can see why you might be thinking sponge for these things, because that does look like a spongy sort of structure. So in that case, wouldn't these grid elements have been mineralised originally?
Okay, this is sort of getting into quite deep areas of the debate, but we know that sponges are quite diverse in their early history. And for example, there are today demosponges which have an entirely organic skeleton.
Okay, does it look like that skeleton?
No, it doesn't.
Okay.
We also know that there are things like the ascosponges, which have a very thick organic layer to the spicules. They would have been mostly, at least, having some degree of mineralisation to the spicules as well. But I wouldn't rule out there being some offshoot in which you had a very different type of skeleton in which the mineral component's been lost and it's doing things with fibres instead.
Okay, so you take something like a leptometis, which is basically a bundle of parallel mineralised fibres, and you take away the mineral and you're left with a bundle of parallel fibres. Is that the sort of thing you're thinking of?
That's the sort of thing I was thinking of, potentially.
I could see that.
What you'd end up with there is not so much a bundle, but rather a thin-walled tube in which these fibres are distributed around it, they're longitudinal. And so when you flatten that, you get what we see in general in the earlier specimens, where it's a sort of an even array of bundles across the distribution of across the width of it. Here's different.
Okay, yes, it is. Also, so, okay, if this was sponge, how would you explain all these fibres that stick out at the top? Because that's one of the really consistent features among all the specimens.
It is, and many sponges have marginalia, which are spicules that project around the top, surrounding the osculum, which is the exhalant hole at the top. It's thought to be to stop sediment getting in mostly. And in this case, it's not so much a thin wall we'd be looking at as a very narrow sort of cavity down the centre, which is defined by these obvious longitudinal fibres. The rest of the wall would probably have been solid soft tissues to some extent, with these little elements within them. And the osculum would actually have been very narrow, and where those little fibres coming out the top.
That's not impossible for a sponge, is it? But so then we've, in that case, we might have three species of this thing. We've got the long, thin one, we've got the fusiform one that's a bit thicker in the middle, and then this one's a different shape again. It's widest at the top. One possibility is that maybe this one's a sponge. Maybe the other ones are algae. What we don't know is whether the fibres are the same thing.
We don't. But we do know that there is nothing like those fibres in any described sponges or in any described complete body fossils of anything else, really. We just know that there are masses of these at this time period in this basin.
That's true, but then, given you've only got one specimen of this shape, could it be that the thing sticking out the top is just the sponges overlying something like a retiolitid graptolite, perhaps?
A retiolitid graptolite?
Or a genuine chitinous hydroid, which do exist.
Right, okay, so one specimen is always a problem. And it's very conveniently in the midline. We would also have to look at the fibres next to it. We can zoom in and see whether in the rest of the specimen there are also lots of little fibres, short lengths of them.
And that would be helpful, wouldn't it?
It would. Let me brighten this slightly.
Okay, so we're looking for reasonably thick fibres, relatively.
Well, we're looking for fibres at more or less the same thickness as these ones in the middle.
Okay.
And there's a diagonal one going across just below the surface here.
I see that, but if this thing is overlying something like a retiolitid graptolite, that can be part of the same graptolite.
Okay, so
it's not one somewhere else in the specimen.
Okay, let's have a look further round.
Let's go down a bit. That's still whatever it is.
There are lots of bits you're passing over.
Okay, stop me when you see one.
Okay, back up a little bit. Stop. So top left here, there are quite a few little black fibres.
Yes.
Yeah,
Yeah, there's one nearly out of the field of view there. That's reasonably convincing.
yeah, there's actually,
Yes.
once you get your eye in, you can start seeing there's little fibres everywhere.
Okay, they're much shorter, these fibres, though, aren't they?
They are. But
I think it would be really helpful to be able to see the microstructure of these fibres and see if they're all the same thing.
it would.
We'd need scanning electron microscope for that.
We would, which we don't have here, alas.
Let us know, but we might be able to get access to one.
Yeah. But I will pull Lucy up on, she mentioned retiolitid graptolites, and you lot don't know what those are.
Oh, well, you're in for the treat. Shall
What?
we look at the first specimen again?
We probably should, because there was something next to it, very conveniently, which might actually help to give us a direct comparison between a retiolitid graptolite and this one.
And I've got graptolites into this video. Yes.
My humble apologies, I can't stop her sometimes.
Alright, so for those of you who don't know about the amazing creatures the graptolites are, Joe did do a video a little while ago, so that's well worth watching. And watch all his other videos as well. And don't forget to like and subscribe, please. Was that what you wanted me to say?
I hadn't thought of such things, but I'm glad you did. Well,
Right, thank you. Yes, so back to the graptolites. So this is a bit of specialist type of graptolite that, you know, we have our normal graptolite like that one there that's got a continuous skeleton. So each of these little right angle bits was a little cup called a theca in which one zooid lived, one or more possibly. And most graptolites have got this sort of solid sheet of, we call it periderm, the skeletal material. It's
organic?
It's organic. Yes, thank you, Joe. It's organic. It's proteinaceous in some way. But some graptolites in the Silurian went a bit minimalist almost, and they reduced the skeleton to a meshwork with just a very, very thin sheet of periderm between the struts of the mesh. And so what you normally get is just the meshwork, and that's what we've got under the microscope here right now. This is probably a Gothigraptus, but I'd want to go and double check that before I put it in a paper. And you see the beautiful sort of elegant lines of that meshwork there. That's absolutely lovely. And it's absolutely totally different from this weird algal sponge, whatever it is, thing to the left. So whatever these things are, they are definitely not retiolitid graptolites. There you go.
Okay. Can we zoom in a bit to have a direct comparison, just the size of the thickness of these branches or the fibres?
Oh,
just
I think...
yeah, just so you can see them both.
Let me fibble. Let me focus. There we are. That's quite nice. So...
yeah,
the struts of the retiolite are that little bit thicker than the fibres of the thing. But I think the struts of the retiolite might actually be about the same thickness as whatever's sticking out of the top of that spongy looking one. So I think if we manage to get these under electron microscope, we're going to want to compare that retiolitid, which is handy beside the thing, with whatever's at the top of the sponge thing.
Yeah, that's reasonable, yeah. And one thing is that these always branch. They are a network. There are no individual fibres within the retiolitid. Is that true?
Yes, they're always a meshwork. I don't think you get any graptolites that are bundles of parallel fibres like that.
Yeah.
You get some that are by bundles of parallel thecae, something like acanthograptids, but in that case, those would be relatively thick tubes, big enough for a zooid to live in.
Yeah.
So again, it's a totally different structure. It's that the tubes would be much, much thicker than the tubes we see in this problematic fossil, whatever it is.
Yeah. So I'd agree we want to check just in case there's anything odd going on there, but the way that those fibres were arranged, coming out the top of that wide one, to me doesn't look like it could be retiolitid.
I don't see it's any type of graptolite at all.
No.
Yeah, we can stop worrying about graptolites now.
Okay, well that's good. So let's try to summarise what we've got here and where it takes us. So we have the first articulated and probably complete, as far as we can see, specimens of whatever it is that produces these banded tubes. An extremely abundant microfossil, not quite micro, but very small fossil that you get washed around in these Silurian seas across large areas of the Welsh Basin, and also other areas as well. So across Europe and North America they've been found too.
Yeah, it would be quite surprising if this was the only articulated material. So you do wonder if perhaps lots of people have one or two specimens sitting in their collections and they're not quite sure what to do with them.
But you would think, because they have had publicity, that anyone who had one of these at home would have published them by now, wouldn't they?
I don't know, I think most graptolite workers have probably found chitinous hydroids and just frankly ignored them because they're not graptolites. Maybe, theoretically, hypothetically.
Okay, yeah, that's theoretically possible, but but don't say that too loudly, I guess. So we can hope that maybe one of you out there has similar things as well, and that would be an important part of the story. We also would like to get back and find more from the site, because the more duplicates you have showing the same structures, the better.
Yeah.
But even with what we've got, I think we can say some interesting things, and it's led to interesting discussion. So first of all, are they graptolites?
Definitely not.
Okay, that's one thing we'll bet.
Could they be chitinous hydroids like all the graptolite workers have been saying all these years?
Don't think so.
Why not?
Because chitinous hydroid skeletons don't, or colonies don't look like that in totality. The other thing is that the individual fibres should have had little cups on them. There is no sign of anything like this at all. They don't form a sort of a series of bundled things in one sort of tubular structure. It's just not anything like chitinous hydroids.
Yeah, there's been a few papers on chitinous hydroids of Ordovician Silurian age over the years, and even from dissolved out material, so you've got very tiny bits. But even in the very small bits, they usually show blanching, which none of those fibres seem to blanch at all anywhere. So I think we can pretty sure they're not chitinous hydroids.
Yep, I think so.
Okay, so that's fine. So that's two things ruled out.
Land plants.
It's looking less likely all the time. The first two specimens we looked at, that might be plausible, but the third one, it's really not a land plant structure, is it?
Yeah, assuming that third one does hold up and it's not two things overlying, which I don't think it is, just it would be so coincidental having the way those fibres suddenly flare out and become irregular just as they leave the body.
Well, could they be algae?
Algae is an interesting one. If they are algae, even algae which have a sort of tubular structure are reasonably delicate as far as I know. Whereas what we're seeing here is that these things were being disarticulated, decayed, and then the fibres are being washed around for long periods and they are preserved abundantly. These are things that do not break down easily at all.
Yeah, and a lot of them are preserved in reasonably deep water mudstones. That wouldn't have been where algae were living because there wouldn't have been enough light for them. So they could obviously, these fibres could, if they were from algae, these fibres could obviously survive a lot of transport.
Yeah, which is difficult to understand.
Yes, which would mean we ought to be seeing a lot more of these algae in shallow water deposits, if that's what they were, because that's where they would have been living.
Yeah.
Yeah.
And you also find places where you have Cooksonia, early land plants, that sort of thing. And we don't tend to see these fibres so much there. It's mostly in the graptolitic facies, out in these siltstones.
We ought to check actually where the paleontologists have been getting these banded tubes from. Are any of those from terrestrial deposits?
I think some of them might be close to it, but then we also need to check that their banded tubes are what are found in these dense assemblages.
That's true. We don't actually know that what we've got are these same thing as these banded tubes yet.
Yes.
Right, so more work to do.
Yeah, that's something we can hopefully resolve with SEO.
Okay, so you want them to be sponges, don't you?
The morphology of the whole structure looks like a sponge. It seems to be a thin or thick wall, depending on the species, flattened onto the rock with an axial cavity that is surrounded by fibres that stick out in the end, as marginalia do. If it is a sponge, though, it would have to be a very different structure, a very different group to some extent, to any sponges that have ever been described.
Which isn't necessarily a problem. We're back in the Paleozoic with lots of weird early things.
Mm-hmm.
But then if they are sponges, why don't we see them in conservat Lagerstätten? Because there's lots of conservat Lagerstätten in late Silurian rocks in North America.
There are, but in North American ones, the Lagerstätten tend to be in shallow water.
Yes, true. So if these things were living in deeper water, you wouldn't see them in shallow water.
Yeah.
So yeah, okay, that could work.
They also may not have evolved until the latest Ordovician into the Silurian. So it might be that all the Ordovician conservat Lagerstätten and the Cambrian ones, they didn't exist then.
That could work, yes.
So you're potentially looking at an entirely new group of sponges.
Oh, so once again, we could do with a bit more material.
Yeah, and we're
Yeah.
trying to think of other options as well. Anything else that could evolve a little bit to create a skeleton of this type. Sponges, with the ascosponges, there's potentially a route where you could get something like this,
Mm-hmm.
but it's difficult to see most other groups doing it.
Yeah, it's totally wrong for something like Palaeontological. Various worms do make tubes, but in these things, there doesn't seem to be room for the worm to live. So I don't quite see that working either, and I can't think of anything else obvious. So perhaps if anyone's got ideas, they could put something in the comments.
Absolutely. But this is what you could call cutting-edge science, I guess, strangely enough. So this is something which has long been an unanswered question. I mean, they've taken centre stage in debates about early plant evolution, these little banded tubes. And even if they are not the same as the banded tubes that have been recorded by paleontologists and looked at with SEM, what we do have is a structure that has baffled people for a long, long time, which is abundant. It's a major component of the ecology of these Silurian seas. It's really quite peculiar that nobody has ever found articulated ones before.
Or they haven't reported them.
Yeah, exactly. Which comes to the same thing. Got to publish or nobody knows.
Yeah, so we've basically got the classic thing in science where we've got an answer, we know what these things look like, and now we just got a whole list more questions.
Yeah.
So,
but we know what those questions are now.
That's true.
Which is good.
Yes.
So next things we're going to do, first of all, contact a friend of ours who is an algal specialist, see if he's seen anything that looks like this structure or he could interpret it as algae. Look some in SEM, see if we can pin down, first of all, whether these are the same types of organism in these different specimens, and secondly, whether these fibres seem to be the same thing as the banded tubes, or whether they match up with something else for that matter.
Yep.
Yeah. And what else are we going to do?
I suppose go out and try and find a few more.
Yeah.
Perhaps have a look through museum collections, see if there's anything sitting in drawers next time we happen to be down in the National Museum.
Yeah. I've got another idea though.
What?
We could do something really crazy and post it on the internet.
What, the internet?
Yeah, and see whether there's anyone, people we don't know even, who have stumbled across something like this before, and it's sitting in a drawer somewhere and they haven't got a clue what to do with it.
So we're basically asking for help from the entire world?
Yeah, that's never going to work, is it?
Nah.
Nah, let's not do that. Fair enough.